Good afternoon. The title of this presentation is International Federation of Clinical Neurophysiology Recommendations on Frequency and Topographic Analysis of Resting State EEG RIMS. The resting state EEG RIMS were described for the first time by Hans Berger, an Austrian psychiatrist, in 1928. He showed an ample oscillation of EEG activity in the posterior areas during eyes closed condition, and he called those RIMS alpha RIMS from 8 to 13 Hz. When the subject opened the eyes or was involved in mental calculation, the alpha RIMS disappeared, and it was possible to see an high frequency oscillation of EEG activity from 20 to 80 Hz and the beta and gamma RIMS. The resting state EEG RIMS are generated and modulated by ascending activated systems and thalamocortical interaction. You can see in the picture the reticular formation with the components of noradrenaline, serotonin, and dopamine. And you can see also that hypothalamic orexin projections modulate the activity in the cerebral cortex, and the same is true for cholinergic basal forebrain projections. An important role in the modulation of cortical activity generating EEG RIMS is related to thalamocortical interaction that you can see in the brain picture. The spatial and frequency analysis of a resting state EEG RIMS can produce biomarkers useful for the assessment of brain function in patients with different brain disorders, from symptoms onset to follow-up. For this reason, International Federation of Clinical Neurophysiology promoted the production of guidelines. You can see the workgroup involved in this action, really smart scientists. That workgroup accepted the electrode montages with lower than 19 electrodes of 1020 system. They encourage the recording of a resting state EEG activity for at least 1 minute to reach statistical power. They also recommended the digital recording of EEG activity for reuse and reanalysis, and the use of source density derivations to enhance the spatial details of the topography of resting state EEG activity. Finally, they recommended the use of more than one electrode as a reference for the recording of resting state EEG activity in order to have a more reliable topographic mapping of EEG activity. After 20 years, there were a lot of tools for the analysis of EEG data. So the International Federation of Clinical Neurophysiology promoted an update of the guidelines for the recording and analysis of resting state EEG activity. You can see here the workgroup involved in the production of those guidelines. The main issues were the kind of resting state condition, what high resolution EEG, and when the use of individual frequency bands, And when analyze the data at the sensor or cortical source level, and what kind of techniques for functional connectivity of the brain sources of EEG activity. And finally, when the use of linear or non-linear measurements to analyze resting state EEG activity. I want to mention that the work group was composed by several scientists, but a specific mention to Professor Raul Bazar and Professor Fernando Lopez da Silva, who passed away some time ago. The work group suggested that the proper resting state condition may depend on the neurophysiological mechanisms test in clinical neurophysiology. The recording of EEG during resting state eyes closed condition for 3-5 minutes and then a new session with eyes open for 3-5 minutes are useful to investigate the maintenance of stable vigilance in the patients. The rapid change of the condition at eyes closed and eyes open is useful to investigate the switching of vigilance levels. Finally, recording of EEG at eyes closed for more than 30 minutes is useful to study the progressive transitions among vigilance levels from quiet wakefulness to sleep. The work group proposed that the proper electrode montage may depend on the spatial and clinical issues of the experiments in clinical neurophysiology. For example, for the topographic analysis of a resting state EEG activity, the 1020 system plus 6 additional electrodes may be sufficient. Or the analysis of cortical sources of a resting state EEG, a 1020 system up to 81 electrodes may be required. For the source localization of epileptiform activity, the use of a 10-5 system, for example, at 128 or 256 electrodes, may be necessary to have accurate estimation of these sources of epileptiform activity. The EEG is a complex signal, but can be decomposed in the elementary oscillations at different frequencies. This allows the identification of the individual alpha frequency peak. This peak represents the frequency showing the maximum power in the range of alpha rhythms. On the basis of individual alpha frequency peak, one may define the individual delta, theta, and alpha frequency bands. The magnitude of these oscillations can be represented in the EEG power spectrum. Of course, fixed frequency bands can be used in experiments in clinical neurophysiology. However, the work group was not able to identify the best solutions, the best frequency bands in delta, theta, alpha, beta, and gamma. The review of the literature showed that there were several suggestions about the frequency bands from International Federation of Clinical Neurophysiology and the International Pharmacoag Society. And you can see in the table that you can have different frequency bands and subbands for alpha, for beta, and for gamma in the proposal of International Federation of Clinical Neurophysiology in 1999 and in 2017, and also in from the Society of Pharmaco-EEG in 2012. So the use of fixed frequency bands remain an open issue in clinical neurophysiology and needs more discussion for consensus. The workgroup faced the issue of head volume conduction effects, challenging the spatial analysis of resting state EEG rims. It is well known that EEG activity, for example, recorded in the electrode B and A, can be used to study the interdependence of the EEG activity in relation to possible underlying functional connectivity in the cortical areas. However, in this case, the EEG signals recorded in the electrode A and B are mainly due to the same source in the cortical areas. So one may take erroneous conclusions about the connectivity and the interdependence of this EEG activity. Another typical case is illustrated in the right side of the picture. You can see an EEG activity recorded in the electrode B. One may suppose that this activity is related to a cortical source underlying the electrodes, but this is not the case in the example. In reality, is a cortical source tangential to the scalp surface to generate the EEG activity at electrode B. The workgroup proposed the use of at least two independent techniques for the source estimation from resting state EEG activity in order to have a cross-validation of the source solutions. In the picture, you see a typical procedure to estimate cortical sources of EEG activity solving the inverse problem. We record EEG activity at scalp electrodes, and we have a map of EEG activity, and we want to estimate the cortical sources of this activity, and we have to solve the inverse problem. Several techniques are available to estimate the cortical sources of EEG activity using a fine head volume conduction model, including cortical source space. However, there is no unique solution, and this is why you need more than one technique. Other techniques are available to estimate the radial current density produced by the cortical areas and by surface Laplacian estimation. These techniques are not based on fine head volume conduction model, so they can just provide a spatial filter removing the low spatial frequencies typically related to the blurring of electrical fields in the scalp surface. However, they cannot be considered as estimation of the cortical sources of EEG activity in the same manner of the toolboxes using the solving the inverse solution of EEG. The workgroup also tackled the issue of functional connectivity of EEG activity. In the picture, you see that this kind of techniques can estimate the interdependence of two EEG signals at the scalp level, or can estimate the connectivity between cortical sources of EEG activity. The procedures are based on linear or nonlinear assumptions. The resting state EEG activity is mostly linear in humans, but there are some exceptions in patients with epilepsy, schizophrenia, Alzheimer, other diseases. So nonlinear assumption should be taken into account. The linear assumption of interdependence of EEG activity at electrode pairs is based on the relation between the phases of cortical sources of EEG activity. In the picture, you see, for example, the case of two musicians in phase in their activity to produce a musical performance. However, we have also the case that the source of EEG activity is not related to another source in a linear manner. In the example, you can see two musicians which are able to produce interrelated notes and musical performances, not using a linear relation of the phases of their activities. So ideally, one should take into account both linear and nonlinear interrelatedness of EEG activity. An interesting technique is the synchronization likelihood, which reflects both the linear and nonlinear interdependence of resting state EEG signals at electrode pairs. You can see in the picture that the procedure is based on the measurement of electric voltage in one EEG activity at one electrode. And you see, for example, two measurements indicated by the red arrow and blue arrow. And then after a leg, again, the measurement of voltages at the time indicated by red arrow and blue arrow, and so on. The voltages measured with this procedure can be represented in a multi-dimensional space. In this case, we represented just two axes. And you can see that the measurements can be used to represent trajectories in this space. And the procedure can repeat the measurements in another electrode and obtain trajectories in this space. Then statistical analysis can provide the synchronization likelihood of the interdependence of these two trajectories as a measure of interdependence of EEG signals at electrode pairs. However, synchronization likelihood and other techniques are affected by head volume conduction effects. In this slide, you can see, for example, the common drive effect. In the common drive effect, you have, for example, that the source C is coupled with the source B, and the source C is also coupled with the source A. You see that there is no coupling between the source B and A. When you apply um, techniques to study the interrelatedness of EEG activity at the electrode pairs, you can see, for example, that there is a relationship and interdependence between the EEG activity recorded in the electrode B and C, and this is okay because corresponds to the connectivity between the sources. But you can also obtain some fake solutions. We have also the cascade flow case. In the cascade flow case, for example, the source C is coupled with the source B, and the source B is coupled with the source A. But there is no coupling between the source C and A. When you use the techniques to estimate the interrelatedness of EEG activity at the electrode pairs, you can obtain true solutions corresponding to the source connectivity, as in the case of the interdependence between signals recorded in the electrodes C and B and A. But you can find also some fake solutions. For example, the solution between electrodes C and A. This doesn't correspond with the connectivity in the source space. So the techniques using the source estimation may partially solve the issue of head volume conduction effects. However, you can have some erroneous connectivity solutions also with these techniques because you don't have a unique solution when you have to solve the inverse problem of EEG from scalp EEG to cortical sources of EEG activity. So the use of all these techniques need more validation and more consensus about the best procedure to estimate the functional connectivity from resting state EEG activity. In conclusion, linear frequency and topographic source analysis of resting state EEG RIMS are major for clinical research according to the outcome of the Workgroup of International Federation of Clinical Neurophysiology. Nonlinear frequency and connectivity analysis of resting state EEG may require more consensus about the best practice in clinical research. So there is the need for international scientific initiatives to address main challenges, including the optimal choice of linear versus nonlinear measurements for the source estimation or source connectivity estimations, and also how to use the graph theory measures to model the topology of the functional connectivity from resting state EEG activity. This requires more consensus, more validation. So the work group encourage further discussion about these procedures and writing new position and white papers on the use of resting state EEG rinse in clinical research and also in clinical practice. You can see here the people of my group, and they helped me in the preparation of this talk. And you can see here the European funds supporting this research. Thanks for your attention.